Imagine if autocorrect changed just one letter in a text message. I love you could become I lose you. Totally different meaning. Something similar happens in your DNA. A single change in your genetic code can alter a protein, which can change how your body works, sometimes in dramatic ways. Welcome to Sciesmic. I'm Matt, and today we're exploring mutations, changes in DNA that can be harmful, helpful, or completely neutral. Mutations are the typos in the instruction manual of life. They're how evolution happens, how diseases occur, and how you become uniquely you. By the end of this video, you'll understand how a microscopic change in DNA can ripple out to affect an entire organism. Let's decode the mystery of mutations. Before we talk about mutations, we need to understand the amazing connection between DNA, genes, and proteins. Your DNA is like a massive instruction manual made of 4 chemical letters, A, T, G, and C. Genes are specific sections of DNA that contain instructions for making proteins. Think of genes as individual recipes in the cookbook of life. Each gene tells your cells how to make a specific protein. Here's how it works. DNA gets transcribed into RNA, which gets translated into proteins using the genetic code. We call this whole process the central dogma of biology. Every 3 DNA letters, called a codon, codes for 1 amino acid, and chains of amino acids fold up to make proteins. Proteins do almost everything in your body. Enzymes speed up chemical reactions, structural proteins give cells their shape, antibodies fight infections, and hemoglobin carries oxygen in your blood. Proteins are the workers that keep you alive. So here's the crucial connection. DNA codes for proteins, and proteins determine how your body functions. Change the DNA, and you might change the protein. Change the protein, and you might change how the organism works. That's where mutations come in. They're changes to the DNA sequence, and depending on where they occur and what they change, they can have big effects or no effect at all. Mutations are simply changes in the DNA sequence. They can happen in several different ways, but let's start with the most common: copying errors during DNA replication. You see, every time a cell divides, it has to copy all 3 billion letters of DNA in the human genome. Even though cells have amazing proofreading systems, mistakes occasionally slip through. Maybe 1 letter in every billion or so. That's incredibly accurate, but with so much DNA, errors just happen. Environmental factors can also cause mutations. UV radiation from the sun can damage DNA in your skin cells. Certain chemicals can alter DNA structure, and even natural processes in your cells sometimes damage DNA randomly. Your cells have repair systems that fix most DNA damage, but some mistakes still slip through. These unrepaired changes become permanent mutations that get passed on when those cells divide. There are 2 important categories to focus on. Somatic mutations happen in body cells and only affect the individual, like mutations in skin cells that might lead to cancer. Germline mutations happen in reproductive cells, like eggs or sperm, and can be passed on to offspring. Remember, genes are located on chromosomes, those X-shaped structures you've seen in your science class. Mutations are changes to genes on these chromosomes, which means they're changes to specific protein instructions. Most mutations are random, just accidents of chemistry and mistakes. But some factors increase mutation rates, like exposure to radiation or certain chemicals called mutagens. Let's look at the main types of mutations and how they affect the genetic. We'll use a simple analogy. Imagine DNA as a sentence made of 3-letter words. Substitution mutations replace 1 DNA letter with another. It's like changing the sentence, "the cat ate the rat," to "the cat ate the bat." 1 letter changes, 1 word changes, but the sentence still makes sense. The DNA ATG, CGT, ACG might become ATG, CAT, ACG. Substitutions can have 3 outcomes. Silent mutations, where the new codon still codes for the same amino acid, like a synonym in language. Missense mutations, where you get a different amino acid and the protein changes slightly. Or nonsense mutations, where the new codon means stop and the protein gets cut short. Insertion mutations add extra DNA letters. This is like adding a letter to our sentence. "The cat ate the rat" becomes the cax, tat, eth, era, t. Right, everything after the insertion gets scrambled because the 3-letter reading frame has shifted. Some scrambling deletion mutations remove DNA letters. Same scrambling problem. "The cat ate the rat" becomes the atta, tat, at, after deleting the C, right? So these are called frame shift mutations because they shift the 3-letter reading frame for those amino acid sequences. Deletion mutations remove DNA DNA letters. The same scrabbling problem. The cat ate the rat quickly becomes the rats after deleting the letter C. These are called frame shift mutations because they shift the reading frame of those 3-letter sequences. Frame shift mutations, either through insertions and deletions, are usually more harmful than substitutions because they scramble everything else downstream of it. It's the difference between one typo versus everything being misspelled from that point going on. Mutations can also vary wildly in size, from single letter changes to entire genes being deleted, duplicated, or even rearranged. Generally, bigger changes have bigger effects. Some mutations are harmful because they create proteins that don't work properly. Sickle cell disease is a perfect example of how one tiny DNA change can have major effects. In sickle cell disease, a single DNA letter changes in the hemoglobin gene, the codon GAG becomes GTG. This substitution causes one amino acid to change from glutamic acid to valine at position 6 in the hemoglobin protein. That one amino acid change alters how hemoglobin proteins fold and ultimately stick together. Instead of staying separate, the abnormal hemoglobin molecules clump up together, distorting your red blood cells into a sickle or crescent shape. These misshapen cells can get stuck in blood vessels, causing pain, organ damage, and so many other serious problems. Cystic fibrosis results from mutations in a gene that makes a protein channel. The broken channel causes thick mucus to build up in lungs and other organs. Huntington's disease comes from a mutation that creates a toxic protein that damages brain cells. You might wonder why harmful mutations don't disappear from populations. Sometimes they persist because they're recessive. People can carry one copy without any symptoms being shown. Sometimes, like with sickle cell, the mutation has both costs and benefits. Cancer develops when multiple mutations accumulate in genes that control cell growth and division. Each mutation damages the cell's normal controls, eventually leading to uncontrolled growth. Now, here's something that might surprise you. Mutations aren't always bad. Some mutations actually help organisms survive better. These beneficial mutations are the raw material for evolution. Lactose tolerance is a great example. Most mammals lose the ability to digest milk sugar, lactose, after infancy because the gene that makes lactase enzyme shuts off. But some humans have a mutation that keeps this gene turned on throughout their life. This mutation arose in dairy farming populations and spread because it was beneficial. People with this mutation could get nutrition from milk even as adults. Today, about 35% of humans worldwide have this helpful mutation. Now, remember when we talked about sickle cell disease? Here's the twist. Having one copy of the sickle cell mutation, called being a carrier, actually provides protection against malaria. The malaria parasite can't reproduce as well in cells with some sickle hemoglobin. This is why the sickle cell mutation is common in regions where malaria is prevalent. It's both harmful and helpful, depending on the dose. Mutations in bacteria can create antibiotic resistance. While this is bad news for fighting infections, it's beneficial for the bacteria. They can survive when the others don't. This shows how beneficial really kind of depends on the perspective. Some people have a mutation that makes them resistant to HIV infection. The mutation affects a protein on cell surfaces that HIV uses to enter the cells. No functional protein means the virus can't get in. Now, that's definitely a beneficial mutation. Humans have artificially selected for beneficial mutations in agriculture. Mutations that increased corn kernel size, created seedless fruit, or gave chickens more breast meat were all beneficial from a human perspective. Here's the most important fact about mutations, though. Most of them are actually neutral. They have no effect on the organism whatsoever. Now, that might seem surprising, right, after everything we just covered, but there are several reasons why. First, remember the genetic code has redundancy, meaning things happen over and over again. Multiple codons can code for the same amino acid. A substitution might change the codon from GGA to GGU, but both code for glycine. The protein doesn't change at all. These are called silent mutations. Second, most of your DNA doesn't code for proteins. About 98% of human DNA is non-coding. Mutations in these regions usually don't have an effect because they're not changing protein instructions. It's like making typos in the margins of a recipe. That doesn't ultimately affect the dish. Third, some parts of proteins are more important than others. A mutation might change an amino acid, but if it's in a region that doesn't affect protein function, like changing a word in a sentence that doesn't change the meaning, there's really no effect. Scientists distinguish between synonymous mutations, meaning no amino acid change, and non-synonymous mutations that do have an amino acid change. But even many non-synonymous mutations are neutral if the new amino acid is chemically similar to the original. Neutral mutations accumulate steadily over time and create genetic variation without affecting the survival of the organism. The molecular clock helps scientists estimate when these species species diverged from common ancestors. Mutations are the ultimate source of all genetic variation. Without mutations, every organism would be identical clones, and evolution couldn't happen. Now, here's how it works. Mutations create variation. Different versions of genes. Natural selection acts on that variation. Beneficial mutations then spread, harmful ones are eliminated, and neutral ones just drift randomly. Over time, populations evolve. The mutation rate matters for evolution, though. Too few mutations, and there's not enough variation, really, for natural selection to work well. Too many mutations, and the population gets overwhelmed by these harmful changes. The actual mutation rate is just right, low enough to maintain function, but high enough to generate useful variation. Major evolutionary innovations all started with mutations. The gene duplications that created complex eyes, the changes that allowed fish to breathe air, the mutations that gave birds feathers, all began as random DNA changes that happened to be beneficial. Every human has dozens of new mutations not present in their parents. Multiply that over thousands of generations, and you get the genetic diversity we see today. Those differences in height, eye color, disease susceptibility, they all trace back to historical mutations. Sometimes evolution maintains variation rather than eliminating it. The sickle cell mutation persists because heterozygotes or carriers have advantages in malarial regions. This balancing selection keeps both the normal and mutant versions in the population. You can think of mutations as nature's experiments. Most experiments fail because they're harmful, some don't matter because they're neutral, but occasionally one succeeds spectacularly and they're beneficial. And that's how life evolves and adapts over time. So now let's tie this all together. Genes on chromosomes contain DNA, instructions for making the proteins. Mutations are changes to those DNA instructions. Because proteins do the actual work in organisms, changes to proteins can affect how organisms function. Mutations can be harmful when they create non-functional or toxic proteins. They can be beneficial when they improve protein function or create new useful traits and variation. But most mutations are neutral, meaning they have no significant effect on that organism. Whether a mutation is harmful, beneficial, or neutral often depends on the context. The sickle cell mutation is harmful in 2 copies, beneficial in 1 copy in those areas with malaria, and showing that the same mutation can have different effects elsewhere. Mutations explain both genetic diseases and evolutionary adaptations. They're the reason you're not identical to your siblings, why bacteria became antibiotic resistant, and why Darwin's finches have different beak shapes. Understanding mutations is crucial for modern medicine and biotechnology. Who knows, maybe you'll become a genetic counselor helping families understand inherited conditions, a researcher developing gene therapies, or a doctor treating genetic diseases. We're entering an era of personalized medicine where understanding your specific mutations can help guide treatment. Cancer therapy, disease risk assessment, and drug effectiveness all can be tailored to your unique genetic makeup. Now, don't forget to subscribe for more amazing science videos, and let us know down in the comments, what mutation do you find most interesting? Thanks for exploring the molecular basis of variation with Sciesmic. Want to explore more about genetics, DNA, and heredity? Check out our complete science curriculum at Sciesmic.com, where every student can learn, grow, and achieve.